So today we are going to discuss about the remaining drugs or like CNS stimulants. So CNS stimulants, CNS nothing but central nervous system. What is the CNS? Central nervous system here. So CNS stimulants. So some of the drugs they will stimulation nothing but here the stimulation nothing but with the activation, right? So some drugs will be activates the central nervous system. Okay, so here the human body is extremely complicated structure made up of some nearly 5,000 billion cells here. You know, the very large, right, means the length of the cells where that nerve cells. Okay, so in order to enable this large structure of this survive hazards of this environment, so individual cells have become specialized in various functions here. So here the CNS central nervous system it serves to integrate and coordinate the activities of various parts of the body. Right. So here the generally the CNS central nervous system it is two types. Okay. Here CS nervous system is two types here. Central nervous system and peripheral nervous system here. So where the peripheral nervous system which collects information from various parts of your body, and the CNS central nervous system. Okay, so it takes all the takes all the messages which is collected by your peripheral nervous system PNS. Okay, CNS and PNS, and it reaches to your it takes to your the brain. Okay, and that will be right. Your central nervous system analyze that message which is collected, which is obtained from your peripheral nervous system, and it analyze that new messages, and it order to do what to do and what not to do. So that kind of action will be uh, right. So that action will be monitored by your central nervous system here. So here the central nervous system is a very important, right? So it is a kind of mechanism which orders to do the some action according to the means according to the messages is received by uh, means received by your PNS peripheral nervous system. Yeah. Okay. So they are used to increase the rate of respiration in and depression caused by carbon dioxide accumulation in the body. So earlier, naturally occurring alkaloids such as strychnine were used as CNS stimulants here. And nowadays, the currently, the derivatives of xanthine and the caffeine, okay, thiophenylline, so these are commonly used CNS stimulants here. Right. So here the caffeine is more active and widely used alkaloid in the world, and usual doses is 60 to 200 mg. And the caffeine may lessen fatigue and increase mental alternates here. Okay, and it stimulates respiratory center, and therefore it is used to analeptic also. The caffeine is also used in conjugation with aspirin, combination with aspirin in various headache remedies. right? So generally means most of the people are having the habit of consuming the tea or the coffee here, right? So where this tea is having the caffeine, the active compound here, where the caffeine is it is acting as a nerve stimulant. Okay, it is a natural product. Okay, it is a natural product here. And along with this caffeine, we have some other compounds also, which is used as CNS stimulants like xanthine, okay, thiophenylline. So these are the other derivatives which are used as CNS stimulants here. And here first we'll see this caffeine, right? So here the caffeine is found in coffee and tea to extent of 1 to 5%. Right? So here the theobromine is an alkaloid obtained from the coca. So it is methylated by dimethyl sulfate and sodium hydroxide to yield caffeine. So the synthesis of this caffeine can be done by using this particular uh, theobromine here. And this theobromine, when it is undergoing the methylation, okay, methylation in the presence of base alkali medium, it is going to give you a caffeine. And it is interesting to note that where the theobromine is almost devoid of simultaneous stimulating activity, and it appears that one methyl group on the xanthine, another, right, nucleus is essential for CNS stimulant activity. And one more method is available to synthesize this caffeine, where you can synthesize this caffeine by using the methyl urea. So here the methyl urea, when it is condensed with ethyl carboxamido acetate, where you can see that, right? So this is your the 20, right? The serial number here, the 20 in the reaction molecules here. So where it is your carboxamido acetate, right? So in the presence of a base, means here the base you can take sodamide or any sodium ethoxide or sodium methoxide, right? So and it yields to a diamide, and which undergoes cyclization to give pyrimidine derivative here. And further, by reaction of nitrous acid, it yields in nitrosode compound, which is reduced to the diamine. And finally, by condensation with formic acid, right? So we can see that from condensation with formic acid, HCOH is a formic acid here. So it converts into diamine into 3-methyl xanthine. Okay, right? So where it converts into 3-methyl xanthine here, and which is on further methylation with dimethyl sulfate, uh, and it is in the presence of alkali basic medium. So it gives rise to a caffeine here, right? And generally this method is developed by True, right? So True, he was a scientist who is in developed this particular caffeine synthesis, and it is commercially used, means commercially applying production method here. Next, yes, next anti-helminthics. So here the helminthics means it's the worm infections. Okay. So here is a anti-helminthics. These are the worm infections involving parasitization with cystodes. Cystodes means tapeworms here. Okay, generally the tapeworms it is frequently found in the pigs, yeah, and the nematodes, cystodes and nematodes here. So nematodes, it is roundworms. Okay, the roundworms will found in the humans, and tapeworms found in the where it is in the animal like pigs. Okay, and nematodes here, right? So the infection is more prevalent in areas where there is a poor sanitation and poor family hygiene here, and crowded living conditions, and malnutrition. Right. So where this particular the infection of these tapeworm infections and roundworm infections. Okay, nematodes, and cystodes infections. It is very common in the uh, poor sanitation, where the areas with the poor sanitation, ugly places, and in the poor families where they are not maintaining the hygienic conditions here. Okay, they frequently will use this infection. They will infected. Okay, they will infected with these uh, worms. Okay, and here the anti-helminthies. So here the anti-helminthies, which are the drugs which is used to cure the particular these helminthies. So in the plant kingdom, sorry, in the animal kingdom, we know that where there are some phylums. There are 9 phylums are there. Okay, uh, Annelida, Arthropoda, okay, mollusca, and platyhelminthes, nematohelminthes, right? So these are the platyhelminthes, nematohelminthes. So all these are the some kinds of the animal kingdom phylums here. Okay, uh, microbes. So here is a anti-helminthes. These are nothing but the drug compounds which are acting against your the infections of your helminthes. Helminthes will nothing but your tapeworms, roundworms. Okay, so these drugs compound, they will means they will prevent or they will kill this tapeworms, roundworms infections, or they will stop these particular infections here. Yes. And they are toxic compounds acting directly on these parasites here, because these helminthes or right arthropoda, mollusca, all these are some parasites. Okay, yes. And the synthesis, the preparation of this particular anti-helminthes, generally they will prepared by these chlorinated hydrocarbons, phenol derivatives here, and some dyestuffs, and piperazine derivatives, and malarial drugs, alkaloids, and its natural products are used in as a anti-helminthes here. Okay, so these are some kind of the structural features of these particular anti-helminthes here. So in that we'll see the first example is this mebendazole. Okay, mebendazole, where this the mebendazole is a drug which is acting as a means anti-helmintic drug here, and the synthesis of this mebendazole is very simple by involving various simple steps, right? So you can see where the preparation is done by using the uh, yes. So benzyl chloride here. Okay, so benzoyl chloride. Okay, benzoyl chloride, when it is treated with this aniline in the presence of zinc chloride, right? So it is giving you a benzophenone, some kind of derivative here. Okay. Where you see this aniline is heated with the benzoyl chloride, in the presence of zinc chloride to obtain the benzoyl derivative. And further, when it is undergoing the nitration, okay, so when it is undergoing the nitration, so it gives that 4-benzoyl to nitroaniline. Okay, so upon nitration, where the nitro group introduction of the nitro group is taking place. Okay, so we are going to introduce the nitro group at to ortho position to this particular amide bond here. Okay, so then further when we are treating, right? So when we are treating with some reagents, so it is giving you a 4-benzoyl to nitroaniline here, and then find by doing the reduction with HCl and tin chloride, or you can use a palladium carbon. So means for reduction, right? So reduction of the hydrogenation. So you can use the palladium carbon or the HCl tin metal here, tin chloride. So it gives orthophenylene diamine. Okay, orthophenylene diamine here. And further, which is undergoing that condensation with S-methyl thiourea to give it is a 2-amino to benzoyl benzimidazole. Right? So where the imidazole is a diamino mixed nitrogen at 1,3 positions here. If the two nitrogens are in the heterocyclic ring, if the two nitrogens are adjacently or 1,2 means 1,2 right position, so it is called this pyrazole, and when it is at 1,3, so it is called the imidazole here. So we are getting this particular imidazole. Right, so benzoyl diimidazole, and finally it is giving the mebendazole here, right? So finally, when it is treated with your particular chloroacetic acid, right? So chloroacetic acid, so it is giving you a mebendazole drug compound. And it is a broad spectrum anti-helminthic here, right? So broad spectrum anti-helminthic means it is acting against all kinds of helminthics. Broad spectrum anti-helminthic means where the drug compound, okay, so it is active against all the helminthics. Like we have various helminthics like tape nodes, right? Nematodes, yeah. So like that, and it is especially useful in the treatment of human infections. And this particular uh, mebendazole, it is effective against many other nematodes and cystodes here. Next, antifilarials here. So antifilarial filaria, where right, so where it is preventing antifilarials treat uh, treat the infections caused by the mosquitoes here. Right. So under this particular example, we have this diethyl carbam, sorry, diethyl carbamine citrate. Right? Diethyl carbamazine citrate here. So its chemical name of this diethyl carbamazine citrate is methyl 4-methyl 1 carboxamide dihydrogen citrate here. And it is in the filariasis and synthesis of this particular diethyl carbamazine citrate is which begins with by taking the piperazine here. So where this piperazine is acylated with diethyl carbamide chloride. So diethyl carbamide chloride, when it is undergoing the acylation reaction, and further when it is undergoing this methylation, acylation followed by the methylation. So we got this uh, at fourth position, fourth nitrogen, right, substituted here. Yeah, the substitution of the particular moiety which takes place at fourth nitrogen, one of the, because it is symmetrical one. Piperazine is the symmetrical moiety or the molecule. Yes. So where that particular acylation reaction can be takes place either on any one of the nitrogen. And further by treating with formaldehyde and formic acid. Right? So formaldehyde and formic acid. And the purified compound is treated with an equimolar quantity of citric acid. Uh, your diethyl carbamide, carbamazine citrate. Okay. Next, the next important topic is where you know the HIV. It is a very oh, right. So it is very dangerous disease caused by the human immunovirus AIDS. We call the AIDS here. Okay, HIV is a human immunodeficiency virus, and it is generally it is an example of retrovirus. So retro nothing but the reverse, right? So where the when we mention the retro means it is a reverse one. So when we call the retro, when that the river where the virus is the reverse. So here the reverse in the sense generally in the normal, right, in the human being where the that particular DNA formation takes place from RNA to DNA, mRNA to DNA takes place. That is called the transcription here. But in this particular virus, HIV virus, where the reverse DNA to RNA formation takes place. That's why it is called the retrovirus. Okay. And it is caused a condition called the AIDS here. AIDS nothing but the abbreviation is acquired immunodeficiency syndrome. And there are two types of AIDS, like one is HIV. It is responsible for the AIDS in America, Europe, and Asia here. And where the HIV-2 is leads AIDS mainly in the Western Africa. Right? And you know that the world is struggling with the issue from last past 3 decades. And it the treat to treat this disease uh, identified uh, uh, like prolonging drugs only, but they are not curing the completely curing the disease as well. Okay. So there are two possibilities to use antiviral HIV drugs act against two targets. They are the viral enzyme transcriptase and the proteases here. Okay, so there is a need to develop effective drugs against new targets which should have knowledge about the HIV structure and its life cycle. Right? So if you know the structure and the life cycle of this HIV, then it is easy to understand the disease right? Symptoms and the disease uh, progression, right? And the disease infections and treatment of this particular disease. So we'll see the structure and the life cycle of virus. So here the HIV is an RNA virus which contain two identical strands of plus RNA, and within its capsid along with the viral enzyme, so ribosomes, tryptase, and integrase, and as well as other protease pieces. So these are the some what is that the parts of this virus here. It have the P24 uh, capsules here, P20, right? So P25 capsules and GP, right? GP 120 and GP41. So these are the binding sites of this, the protein binding sites of the virus here. So you can see the structure of this virus here. So where it is having this uh, glycoprotein GP41, glycoprotein means dot-me glycoprotein GP120, and RNA genome here, right? And lipid envelope and protease, host proteins here, capsids, and reverse transcriptase enzyme. So all these are the previous parts of your virus. And the website. So the in a, they are bind with host cell, starts reproduction and cause infections here. And HIV, it can infect many types of where it is mostly infect the cells in the human system. Once infected, a cell it can be copied hundreds of HIVs, right? So the HIV copies here means the cells. You see, the several kinds of immune cells have been proteins on the surface. Okay, you see the structure of this HIV virus. So you have this various proteins cells like CD4 receptors and right, GP120 and GP41. So all these are the receptors, the binding sites with these proteins here. Right. So they will binds with the host cell, and they will basically this HIV. It is infects your immune system. Okay, so you know that the immune system is the defensive mechanism. Okay, so it is a defensive mechanism to the body where it is preventing it is preventing to attack the entering this what is it, antigens. Okay, so when the antigens are trying to enter or the attack the body, so the where the antibodies are produced here. Okay, when the antigens nothing but the pathogens here. So when they are trying to attack or they enter in the means human body here, so they will produce the immunity system or the self defense mechanism. It is producing the antibodies to fight against this particular antigens here. And these antibodies are it is produced by your lymphocytes, T lymphocytes here. Okay, so your HIV, it is targeting your, okay, it is targeting your T lymphocytes, and here the T4 cell is responsible for the warning host immune system. And okay, so once HIV binds to an immune cell, so it hides its DNA inside this particular cells of DNA. Right? So this turns the cell into a sort of HIV factory, so it can make more copies of itself. Now we'll see the steps. It's the steps which is uh, occurring during this particular HIV life cycle. So we'll see the first the binding. So binding here the HIV, it contains GP120 and GP41. In tower like right, so in tower like membrane envelope of these 17 proteins here. So GP41 it transfers the envelope and it's bound non-covalent to the GP120. So which projects from the surface. So when the virus approaches this host cell, so GP120 interacts and binds with the transmembranes, a protein called CD4 and the receptors on T4 cells here. So when HIV binds to the CD4 and receptor, so other proteins on this cell surface gets activated and it is allowing HIV to and fuse to enter this cell. So here when this particular HIV, right, so cells are entering into the host cell here, then the reverse transcription, right? So upon means after fusion means entering, uh, right? So when the fusion is complete, means entering into the host cell is completed. So here the HIV nucleocapsid so enter into the host cell and opens its by an enzyme called protease. And then viral RNA and it is related to enzymes released into the cytoplasm. And generally the viral RNA is not capable of coding directly. So to overcome by transform to DNA by incorporation to the host cell, host DNA. Right? So the conversion of DNA into RNA into DNA is not a process present in this to host cell due to lack of enzymes to catalyze here. So HIV carries its own enzyme to complete this conversion. It is called the reverse transcriptase enzyme. And generally, we call this process the reverse transcription here. And then the new DNA is formed. We call this the proviral DNA. Next, integration. So integration is a multiplication here. Right? So integration is a multiplication. So after doing this particular entering into the host cell and reverse transcription, so where the multiplication of this HIV virus is takes place with help of this integrase enzyme. Next, transcription. So we know that transcription and translations, right? So where the transcription nothing but where the formation of the new DNA, right? The new DNA stands here from mRNA, and the translation is formation of the protein here. So since mRNA carries code for synthesis new viral proteins, so each section of this mRNA relates to synthesize a different part of HIV. And full stand-up mRNA is proposed to get transferred or translated into all these viral proteins to multiplication here. Right, so where it undergoes some multiplication and it forms some new viral cells. And the final step where it is a viral assembly and maturation. So the final step where it begins with the assembly of new virus here, where the long string of these proteins translated from this mRNA and then so it is cut up into smaller proteins here. Right, so it cut up into smaller proteins by that particular enzyme called the protease, and these proteins become difficult part of HIV and such structural pieces. You see this, there are capsids and the matrix, so different structural pieces here. We have the various structural geometries here. The enzymes, you see the protease as integrase. Okay, so all these new viral protease gets assembled and generated the new HIV virus from this host cell. The new HIV then takes some time to mature which can go into this infected new cells here. Now we are moving to this HIV drugs. So which the drugs which cures the HIV or the AIDS here. And you see this particular, the HIV drugs are classified into various types like inhibitors of viral reverse transcriptase and the protease inhibitors, integrase inhibitors, and entry inhibitors here. So these are various types of drugs, categories of the drugs here. Some drugs will be inhibitors of viral reverse transcriptase, means the inhibition, right? So inhibition, what is the inhibition? Is the stopping, yeah, the prevention. Okay. So like when, right, particular receptor means drug receptor inhibition here, right? So yeah, the receptor where that it is a natural compound where it is inbuilt of the living organism, and it may be the protein or the enzyme or that carbohydrates or any ion channel here. So these are acting as your particular uh, receptor here, and that natural yeah, the ligand compound. Okay, the ligand compound is going to bind with this receptor active site, and like yesterday we discussed about the enzyme inhibition. There are two types here: the reversible enzyme inhibition and irreversible enzyme inhibition here. So, what are the reversible enzyme inhibition and what are the irreversible enzyme inhibition? And here, the reversible enzyme inhibition again three types like uh, competitive. Okay, competitive reversible enzyme inhibition, non-competitive reversible enzyme inhibition, uncompetitive enzyme. So, competitive means when they are uh, drug compound and the internal neurotransmitter. Okay, exogenous chemical and endogenous chemical here. So both are when trying to entering in this receptor active site, then there is a competition is takes place. We call this the competitive inhibition. And non-competitive means when this particular, right, the enzyme active site, right. So where this receptor means ligand compound or the drug compound, so it is not binding to the receptor active site here. So it is binding to the another active site we call the allosteric site, right? So here the enzymes are having the active site and some additional, okay, the additional active site also present, that is called the allosteric site here. Okay, so it will be the particular ligand compound or the drug compound, it is binding, right? So it is occupying its allosteric site. So obviously there is no competition between the natural neurotransmitter and your drug compound here. And you see that due to, right, so due to binding of this particular ligand compound, I will show what is, for example, here is a, you see this is the active site here, okay, enzyme active site, and it is having, right, so this is active site, this is allosteric site, right, so this is allosteric site, this is the additional or the secondary active site here. So your drug compound is binding with this one, allosteric site. And the binding of this drug compound, okay, ligand compound, binding of this drug compound or the ligand compound to this allosteric site, so it leads to conformational changes of your active site. Conformational changes takes place in the receptor active site here. Okay, so where this particular uh, conformation changes is takes place, so it won't facilitates for this attack of your natural neurotransmitter. In this way, it is inhibiting by entering the particular uh, what is it your natural neurotransmitter. This is one kind of inhibition called the non non-competitive and uncompetitive. Yet uncompetitive nothing but where the receptor means your drug compound or the ligand compound, it is binding to the complex, right? Drug receptor, okay. The drug receptor complex. It is a complex, binary complex, you see. Okay, sorry. Yeah, yeah, engine. Right? In the substrate engine, right? Engine complex here. Substrate and engine complex. And with the drug compound, okay, drug is binding to the substrate engine. Okay, it is binding to the substrate enzyme complex here. So there is no competition because already the substrate is binding with your enzyme active site. Then your drug is binding to this one. So there is no competition between this. So it is uncompetitive. Okay. So like that here, this HIV drugs, right? So they are having the various types like inhibitors, viral reverse transcriptase inhibitors, protease inhibitors, integrase inhibitors, and entry inhibitors. Right? So in this, generally these are the reverse transcriptase nucleoside. Right? In this inhibitors, viral inhibitors, we have two types here. One is nucleoside reverse transcriptase inhibitors and nucleotide reverse transcriptase inhibitors. You know that what are the nucleosides, what are the nucleotides? Yeah, the nucleoside nothing but where the in the DNA, right? You know that in the nucleotide. What is DNA generally? So yeah, the DNA is okay. DNA is nothing but your yeah, here is a sugar unit, delta sugar unit plus nitrogen base and a phosphate unit. Right? So this is a nucleotide, right? This is one nucleotide, and the combination of these nucleotide forms a polynucleotide, and this polynucleotide is forms a DNA. So DNA nothing but your polynucleotide. Okay, and when the sugar and nitrogen base is concerned, it is called the nucleoside. There is no phosphate, right? So the combination of sugar unit and nitrogen base is called the nucleoside. So here the some of the tracks are inhibitors. They are targeting your nucleoside. Some will be targeting the inhibits your nucleotide. So see the nucleoside was transferred. This inhibitors here cannot get. There is a nucleoside. It is analog reverse transcripted inhibitors. It composed the first class of antiviral drug, which is developed here. And in order to be incorporated into the viral DNA, so where the NRDI must be activated in the cell by addition of these three phosphate groups. Today, deoxyribose moiety, and where it is going to form your RTA triphosphate here because the three phosphate units are combining and form attacking on this particular nucleoside. It's going to form your nucleotide reverse transcriptase inhibitor triphosphate. And the partial step is give the phosphorylation. So you see the example under this is zidovudine, where this zidovudine it is having this particular right. So okay, so you see this rate of ribose sugar where it is going to attack with this particular yeah azido group here, and you right? So in the ribose sugar, you can see the having the hydroxy groups here at 50 position. But in place of in this particular okay, preventing to form the what is it? Yeah, DNA polymerization something, right? Because you see DNA formation. So the DNA, how the DNA is forming by addition of the nucleotides. So the nucleotide, one nucleotide and another nucleotide is right the binding with the phosphate, or that they are connecting, linking with the phosphate here. So, but here there is that phosphate connection is done with the hydroxy group. Free hydroxy group is there. Then the linking will be takes place. But there is no hydroxy group at third position in this particular pentose, right? Heterocyclic compound. But instead of that hydroxy, there is a azido group here. So here the azido group incapable to linking that phosphate here, other nucleotide. Okay, so it is going to stop that chain elongation. Right? So with it leads to the chain termination. So here the zidovudin, it is originally developed by an anti-cancer agent. Generally, here the zidovudin, it is also acting as this means it is used for the cancer treatment and it is acting as an anti-cancer. And later it is found that this is also used to treat the AIDS. Okay. And it is a so there you can see there is a thymidine moiety, right? There is a thymidine moiety here. And where it has been replaced by the acetopone. Okay. So you can see that there is no hydroxy group at third position. So instead of that, there is azido group here, where this the hydroxy group it is helping or it is for the DNA formation of the virus here. But generally, when it is replaced with the azido group, where the termination of this particular uh, right the DNA chain is takes place. So here you see that unfortunately it can be of reverse side effects here. And you know that every drug is having a few adverse effects or the side effects here. Like this is also Zidovudin also having some side effects such as that anemia. Anemia means with lack of blood. Okay. And some studies of this target enzyme has allowed to develop less toxic nucleoside analogs such as the lamivudine. So lamivudine means by modification of the structure, the lead modification or the structural modification. So the one more drug which is with the minimum side effects or the less side effects is developed. That is called lamivudine. Next, nucleotide reverse transcriptase engine does. Here we have this example under this example, naviprapine, navirapine, sorry, right? There is a navirapine, which is a drug used to treat and prevent the HIV or the AIDS here. So, and it is generally recommended for use with other antimicrobial, sorry, anti retroviral medication. It may be used to prevent mother to child spread during the birth. And we have the protease inhibitors. Under this, we have this indinavir and nelfinavir. Okay, indinavir, nelfinavir, and integrase inhibitors. Right? So these are some types of drugs, and entry inhibitors. We have this enfiradine. So these are the some drugs which are prevent or the few the HIV.